Hi, Victoria. So nice to see you here.
Thank you. Hi, Katarina. So nice to be here, too.
How are you feeling?
I'm feeling a lot better, thanks.
I'm so glad to hear that.
Yeah, me too. Not perfect, but a million zillion times better than before.
Hi, Vincent. How are you today?
Ooh, Vincent, your PTR is beautiful.
Thank you so much. It's actually one of my photos and NFT of mine. I don't seek to make NFTs like the rest of everyone. I like making one-offs that, have inherent value themselves, and the utility comes with having it. But
Hi, Leo. Oh, sorry. Go ahead.
I only went into the Web 3.0 space as an exposure campaign to show that I can enter a realm of business in which I hadn't operated in before and create wakes. I didn't know they were going to be small, large, but 400,000 NFT creators, I guess, dropped out, and there's only roughly 15,000 that are still even bothering because they're the only ones still potentially selling. I have yet to even price mine yet.
Well, that's great. Hi, Leo. Welcome to the
Yeah,
room. Welcome to Clubhouse. We're glad to have you here. How's your day?
Hi, can you hear me?
barely.
Yeah, I can hear you well.
Oh,
But Vincent can't. I don't know. Maybe try again.
Well or barely?
no.
Could be a little clearer, perhaps. I don't know. Sometimes when people wear headphones, instead of just using the speaker, it gets a little bit muffled. So
Yeah,
you might.
no, I'm not wearing any headphones or anything. It's just my Mac. So I'll
Oh,
just try to be louder, I guess.
yeah, that helps a lot. That helps a
Yeah.
lot. Thank you. Hello, John.
Hi, everybody. Happy afternoon, evening.
Hi, John. Meet Leo, our guest speaker here today.
I'm in the Matrix. Sorry, I'll just be in listen mode till I get out of the Matrix.
You're no longer quantum entangled.
Yeah, we will start in around
It's his name.
8 minutes. Yeah, in the meantime, you're welcome to. I shared the paper link in the chat. You're welcome everyone to check it out, and then we have the presentation pinned on top of the room that we will go through. Leo will go through it in a little bit, but we start on top of the hour. So yeah, thank you for being here, and we're excited. Thank you, Leo, for coming here in Clubhouse and going through all the trouble. We really appreciate it.
Thank you for having me. I mean, this is quite interesting. I just installed the club desk, I guess, today, and so I'm still getting acquainted and familiar with the environment, and yeah. So I have plenty of slides. I mean, what is typically the format of these meetings? Do you want me to kind of briefly talk about the material, mostly leave time for discussion? Do you want me to go into the details?
It's really how much how much time you have. I would leave enough time for questions, but we have time for details but it's more about how much time you have to later then still answer question, because that's a kind of nice thing about this platform, that people can interact with you. So maybe in between, something in between, because I don't know what time it is for you, how much time you have to be here. So it depends more on that, like on your time.
Yeah, so it's I'm in Texas time, so it's almost 8 p.m. and um, I mean I have time, but well, let's see. I'll give you an overview and just the very basics of the technology, and depending on the interest and questions, we may go and dive deeper. And of course, I'm available at the later stage to discuss.
Yeah, sounds perfect. Thank you.
Yeah, I was gonna say agree. It's really your space, Leo, and we're hoping that if people want clarification or you bring up a point that somebody wants to discuss with you, then maybe we'd have time for that. It's really a space for you to just develop your topic in a way that makes you feel like you are able to share the way you want to share and share what you want to share as well. So it can really evolve organically, and up to you.
Okay, thank you.
And we will lead a Q&A, so you don't need to worry about anything like that. And sometimes guests will put a question for you in the room chat that you can see and we can share with you also. So you don't need to worry about that either. And just have a great time sharing your work.
Sounds good.
All right. Well, I have some stuff, some work of mine that I've been working on for quite some time. It's an e-learning platform in which maybe one of the more feature-rich and affordable in the world, of which the next moves are to integrate them with several other MVPs I have, one of which you can feed it a markdown file or textbook, and it will spit out a curriculum, homework, quizzes, and tests. Another module I'll be adding is one of my favorites of all of my MVPs. I didn't create it, it was given to me, but I'm doing some adaptation to it, and it's a ASL to audible speech and written text in real time. It's absolutely wonderful. And this is all for, as I said, education. And also, well, the underlying principle is to give, well, prodigious individuals the attention they deserve, not to leave out people who aren't of prodigious nature, rather, but those who do and excel with no means, or I mean, no, not no means, but rather no other means other than the pursuit of knowledge and their capability to process it. I find that's the reason why we don't produce Einsteins and Galileos and Da Vinci's anymore, because back in the day, individuals were paid to very much train these prodigies so they could, grow to be the next leaders. We're not really doing that anymore. I just got in a room. It was very much an argument. All I'd ask was, does anyone have any role models these days? And I really was asking. I wasn't trying to prove a point or anything. And someone said Elon Musk, and then it broke out into a very much of an argument. And it might have sounded like I was against Elon, only because I'm also of the ASD, and I've been through many experiences he has. We both have lots of financial background. No relationship with our father. Very close with our mother. Very much have felt alienated our whole lives, and just we've gone.
Vincent,
Or-
excuse me, Vincent. I'm sorry. This would be great for you to open up a room and just share all about this right now. We're just about to start the room, so instead of cutting you off later, I'm going to ask you to pause that right now and maybe consider having your own room where you can have this type of discussion. And I'd like to hand the mic over to Katarina so she can start the room and introduce our guest and get things going. Thank you.
yeah, thank you so much. Yeah, this is like a guest speaker invitation room. So I know we also do open discussion rooms, but yeah, let's just start. It's 9 p.m. EST. So thank you everyone for coming, and of course, welcome everyone to Science Society, and a special welcome to Dr. Leo Bleris. And before we start, let me introduce you to the audience so people can get to know you a little bit. And yeah, and then usually Victoria asks a couple of interview questions if that's okay with you, and then it's time for your presentation. Okay.
Thank you. No, yeah, that's absolutely fine.
Okay, then yeah, Dr. Leonidas Bleris, he's at the University of Texas, Dallas, and he is a Cecile H. and Ida Green Chair in Systems Biology Science and a professor with the Bioengineering Department of the University of Texas at Dallas. And before he joined UTD, he was a postdoc at the FAS Center for Systems Biology at Harvard University. And he did his PhD in electrical engineering at Lehigh University in 2006. I hope I said that right, the university name. And he received a diploma in electrical and computer engineering from Aristotle University of Thessaloniki, Greece. And he was awarded the Christine Mirzayan Science and Technology Policy Graduate Fellowship from the National Academy of Science. And he served with the Board of Mathematical Science and their applications. And he won different awards throughout his career, and he is focused on systems biology, mammalian synthetic biology, and genome editing. And yeah, he has received support for his work from NIH and NSF and the NSF Career Award. We are very honored to have you here today. Thank you for coming. And Victoria goes ahead with the interview. Thank you.
Thank you, Katarina. Leo, that was really fantastic to hear about your work. And we are so honored to have you here with us in Science Society today. So thank you. Thank you very much for coming and downloading the app and all that. So my question is just to give the audience a little bit of a background of the guest speaker, which tonight is you. And my question is, if you can think back through your life and consider when you might have felt a special connection to science, and I don't really mean as a career, but something that you noticed that you felt an interest in. And if you can, then if you could tell us a little bit about that moment or event or person, and that could be in childhood or adolescence, really any time during your life.
Yep, thank you for the kind introduction, Katarina, and it's my pleasure to be here. Hopefully, we'll have a good discussion. So, Victoria, my first, I guess this is a nice question. I grew up in
Could you move a little closer to your microphone? Your voice is kind of soft.
yeah, so is this a bit better?
Yes.
Okay, thank you. So so about my trajectory, let me start first with this. As you see, I moved from engineering, from electrical engineering to more molecular and cell biology at some point while I was finishing my PhD. My postdoc experience essentially was my first exposure to wet lab work. I was hired as a postdoctoral scientist at the Center for Systems Biology at Harvard to apply engineering and control theory principles in cells. So they were interested in looking at biological pathways and using theory and knowledge that we have from network theory, from control theory to apply sophisticated control. So in my career, I've always kind of stayed at that interface between sciences. So I find biology fascinating, but at the same time, I'm an engineer. So I look for ways to modify cells, to build new technologies, and to essentially, I try to provide solutions. Now, I grew up in I'd say, academic family. My father was a physicist and my mother a mathematician in Greece, that is. And I don't know exactly where my first exposure to, you know, I guess my first initial kind of what triggered my interest in the sciences. But I remember my father always used to try to model simple things with simple mathematical models, and I'm talking about, your relationships to people. So he would just write a simple mathematical model that kind of captures the energy as the relationship and equates it to some interaction. So simple things like that kind of inspired me, and then I guess it was a they said that from a physicist and a mathematician you can become an engineer, I guess, and I took that path essentially. Yeah, so I can't pinpoint it exactly, but I guess it was mostly my interactions with my father.
Thank you so much. That was a really wonderful talk about your work and your early life with science. And so I'm hearing, it sounds like your father was instrumental in expressing the world to you through mathematical principles in a way. So it was another form of communication that you were shown, like another language, maybe.
Exactly. Yeah.
Yeah.
So ways to model simple interactions, simple decisions. Just every action as a reaction, things like that. So basic physics principles.
What a gift. It really is. What a gift. Because now we get to hear about your work today, but that was the path that your father sort of opened for you with that form of mathematical communication, with that modeling. And everyone has their unique connection, and we really do appreciate hearing from everyone, because they are all so different, and sometimes we may resonate with them, and other times it just leads us to understand how important it is to lead by example. With young people and anybody around us, you never know who's learning from you. But also, you mentioned that the interdisciplinary nature of things. and
exactly.
how
Yeah,
important that is.
I try to do the same in my in my classroom. So I try to move beyond exactly what I'm teaching to my students and talk about the philosophy of things and potentially even poetry, and so how everything is connected in a way. So there are general principles that describe uh, phenomena that you can use and take from one domain and move it to another domain with certain approximations, with certain freedoms, of course.
Sure. I hope that example will be imprinted on the people who work with you, and it's always my hope that conventional schooling will be more like that someday and not regimented into artificially imposed subjects. But I would like to get out of your way now and
Yeah.
pass you the mic for you to go into your talk. And we've already discussed that however you'd like the Q&A to be, driving your discussion or following your discussion, that's up to you. And we have your PDF pinned at the top so people can follow along and you can take us through it. And we're just really looking forward to hearing about your work. So thank you so much. And the mic is yours.
Okay, thank you again. So I said the link of the presentation, so I'll just go through a couple of the slides, and at minimum, what I'd like to do is explain what physical and clonable functions is, how it can be used today, and talk a little bit about CRISPR, um, um, and discuss basically how this technology can help solve some really important problems that we have. So this is work that started a couple of years ago with discussions with another faculty member here in the university. This colleague of mine, Dr. Macris, is an expert in security and security-related applications in the semiconductor industry. And so we discussed about technologies in the semiconductor industry that could potentially be transferred in a way, or we could use inspiration from such technologies to solve problems in biology. So if you go to the second slide, it contains a very basic definition of physical and clonable functions. So physical and clonable functions, or PUFs, are a physical entity, something physical, something manufactured, something that is built, that provides a measurable output. It's something that you can quantify, and then you can use this measurement, this data that you get from that physical entity, as a unique and irreproducible identifier for the artifact or whatever device or whatever host it contains the PUF. So this can be quite complex. So I'd like to move slowly here and try to help you understand the concept of a PUF, as much as possible. So if you go to the next slide, PUFs are popularized, they were invented in the semiconductor industry. So what happens in the semiconductor industry is the companies they build chips, they build molecular gates in individual chips in using wafers. So these are These are silicon plates, and in these wafers they print and they build using semiconductor manufacturing processes thousands of, again, chips on the same wafer. Now, if you zoom in, let's assume for a second that you have a wafer that has 2 chips built, right? Obviously, this is not true. You have thousands, but let's assume that we have 2. And you zoom in these 2, and you look at 2 gates, the silicon gates in these chips. These are illustrated here on this cartoon that I have. Now, because of variations in the manufacturing process, and these have to do with the doping, deposition, photolithography, etching, anything that has to do with actually physically manipulating the material that forms a gate, a silicon semiconductor-based gate, there are small differences in these gates, small, minute differences. You see this, for example, here you have this so-called depletion region that is slightly different between the 2 figures. Now, the result is that at a high level, the gates behave approximately the same. So they're kind of on-off behavior, let's say, when you apply some signal, the response is approximately the same, but it's not exactly the same. And that is because of this stochasticity in manufacturing these chips. So that is a key concept, and the groups in the semiconductor industry realized that potentially, when you combine these gates in a chip, you may be able to get some information that is unique for the particular chip. You may be able to excite, put a specific input, a specific voltage to the chip, and obtain a measurement which is unique, specific to the particular chip. So the 2 chips that I mentioned in the beginning, so that are next to each other, will be level, their on-off behavior will be the, but under specific input conditions, they will give you a unique output. So if you go back to the previous slide, a PUF is a physical entity, right? So it's something that is manufactured, which provides a measurable output. So that manufactured gate, you can apply an input signal and get an output measurement that is a unique, that is, to the 2 chips that I mentioned before are different. They provide, you can get a different readout. And irreproducible, that is also key. You cannot build that chip again, because the way that it was built, it was a process, it was a result of the stochasticity in the manufacturing process. So these small differences in the individual gates resulted into this irreproducibility. So moving on to the 4th slide, PUFs have there are 3 criteria for PUFs. One is that they are robust. So once you build a PUF, you can provide the chip to a customer. The customer can perform the measurement on the PUF and will get the same signature, right? So the signature, the readout from the PUF does not degrade. It's unique. That means that you cannot rebuild it, replicate it. If you have 1,000 chips on the same wafer, each one is different enough, and you can then sell them individually to a customer, and the customer will be able to read out, recognize the PUF, but they will not be able to build it. The unclonable part is, so the unclonability is the last bit, which is important. Again, once a customer has a particular chip, they cannot replicate it. They cannot build a chip with the same PUF. Again, it's impossible because of the stochasticity of the process. So why is this important in the semiconductor industry? It has become extremely important because companies use these PUFs in chips that they manufacture for so-called provenance attestation. So they validate that the chips were manufactured in a specific facility. So that is extremely important because because there's because of potential stealing of IP for verifying that the chip was again manufactured by specific facilities is extremely, it can be extremely important, right? Right now there is interest in the electronics and hardware in terms of hardware security, and it's been it is extensively used. I'm trying to locate the exact number. I don't want to quote, but it's something that in semiconductors now has become that fingerprint that is introduced in the manufacturing process has become commonplace, let's say. So now, if you move to the next slide, what we identified an opportunity that potentially the similar technology could help is with cell lines, with the use of cell lines and with cell line engineering. So cell lines are extensively used today in labs. We use cell lines for studying a disease, for studying biology, for disease modeling. There are cell lines that are used for biomanufacturing, cell lines that are used in bioreactors for production of valuable compounds, for metabolic engineering, for production of antibodies. The industry for cell lines is about $20 billion today, and it's expected to grow to more than double in a few years. Something that is driving the developments in cell engineering is the ability to perform sophisticated edits. So you perform genome editing using CRISPR and introduce mutations, modifications, pathway changes at will, essentially, and again, do use the cell lines for a number of biomedical applications, either a two-dimensional or 3D organoids and so on. So you can imagine that we are at a point right now where we can use cell lines for personalized medicine. So we can basically isolate cells from patients and perform basically do IPS cloning and move from fibroblasts to specific tissue, or we can start from a baseline cell line and introduce mutations that are present in a certain patient, and then use these modified cell lines for disease modeling. Now there are tens of companies right now that sell modified cell lines, and we believe that it is in their best interest that the cell lines that they build are somehow secured, so that you can introduce a unique ID that verifies that the cell line is genuine and that has passed quality control from the company, and that you can indeed use it. That is from the customer side. So the customer will benefit from having that information. The companies on the other side are interested in selling a single cell line to a single customer customer and verifying that transaction. Right? It's commonplace nowadays that cell lines are basically shared among labs and universities. So there's essentially now having a unique identifier will give companies the opportunity to to enforce their IP essentially. So if a company builds a particular mutant and spends resources for it, then they can sell that individual cell line to individual customer and perform provenance attestation. So the paradigm is in slide number 6. So we think that if you see on the top left side, you have a manufacturing and distribution protocol where a company has a valuable cell line. And what it does, it performs inserts that PUF. And again, I have not talked yet about how we do that, but bear with me. So you have a cell line, and in one step, you introduce introduce a PUF. So you produce, let's say, in one step, 10 or 100 clones that carry different PUFs. So these individual clones can be sold to customers, and the customers then, by sequencing a particular location in the genome where the PUF rests, can perform the provenance attestation. So I'll repeat that the concept of the PUF requires 3 criteria. This is the robustness, uniqueness, and unclonability. In this paradigm, robustness means that when a company builds a puffed cell line, so it has a single cell line with a puff, when it gives that cell line to a customer, the customer performs the readout from the cell line, the sequencing of a particular location, and this is simple amplicon sequencing. It's nothing too expensive or time consuming. And so with that sequencing, then the information that the customer gets should be the same that you have in your database. So that is the robustness. So the signature, then it's maintained. Uniqueness means that 2 clones, 2 puffed cell lines that you've built will not have the same signature. So they are unique. They're different to each other. Unclonable means that any customer cannot take the original wild type cell line and introduce a puff at will. It'll be impossible for a customer to actually build a puff and kind of violate any IP. So now, how did we build the PUFs? So we used CRISPR, and we used a couple of tricks, a couple of a two-step process, let's say. So I don't know if I need to go into what CRISPR is, but essentially, if you go to slide number 7, you must have seen something on the news, something in a science journal about CRISPR. It's a genome editing technology that has revolutionized the genome editing field. So the field is not new. It's several decades old, but it's only with the discovery of CRISPR that has really that things have really accelerated, and it's because of the ease of use and the efficiency of the technology. So before CRISPR, the researchers, including myself, we used engineered proteins that were not as easy to build and did not work as well. So these were zinc fingers or TALs. But anyway, so with CRISPR, things have simplified significantly in terms of genome editing. You can go inside cells and perform individual single nucleotide edits. The technology resulted into the Nobel Prize last year in medicine for the group from Berkeley. and Munich, the two ladies. And obviously there's Feng Zhang from MIT that led a million developments. So if you go to slide number 11, essentially the technology relies on two components. So you have a protein, CRISPR-associated protein, which we call here Cas9, which is that gray kind of pulp. And you have a guide RNA, that gRNA, which is an RNA sequence that associates with Cas9, with a protein. It creates a complex, and you can use that guide to direct the protein into any location in the genome. You direct the protein to any location in the genome by selecting that 20 nucleotide target sequence. It's the red, highlighted with red sequence. So these 20 nucleotides, all you have to do is select them so that they're complementary to a target, to a desired target in the genome. So if you have a location in the genome that you want to edit, all you have to do is select these 20 nucleotides and then clone them within that guide RNA structure, which contains a particular, it's a special sequence, so it contains a hairpin that is then recognized by the Cas9. Now, when these two come together, they essentially very efficiently find the target, and they can introduce a cut. So they can introduce a double-stranded break in that particular location. So that works quite efficiently. I mean, it obviously has its issues, but it works quite well. Now, once you cut, the cell will try to correct the damage. There are ways that you can intervene, that you can hijack this process and insert new information, or you can let the cell fix the error, fix the damage by itself. And that process, when the cell fixes the damage by itself, it is stochastic. It is called non-homologous end joint repair. And so if you go to slide number 12, which is the next slide, it shows the result of that random fixing of a cleaved sequence. So if you look at this set of sequences, and I'll pause here because this is quite important. This is the source of stochasticity that we use to build PUFs. So if you look at the sequence, this is the result after a cut. So before cutting, what we had was the top sequence. The top sequence is the one that has the blue string of letters, of nucleotides. So that cell line, we had thousands of cells on a plate, and then all the cells had that same information. So we built CRISPR to attack that particular sequence that it is blue and red next to it. So the CRISPR was designed to cut between these two A's, the blue A and the red A. So after we cut, we let the cell fix itself, and then we sequenced the cell line. And what we got was essentially about 13%, if you see on the left, of the original sequence. This was uncut, basically, cells. Then the next one is 12%, and you'll see that there is a gap there. That gap, essentially, it's after you align the two sequences, you have a gap. It means that that 12% of the cells are missing 5 nucleotides right in the middle. So it's a continuous sequence, but it's missing 5 nucleotides. The next 10% misses 6 nucleotides, 9.5% of the cells miss 3 nucleotides, and all of them are at a different location, as you see. So it's random. right? So once you cut in the particular location, you have random deletions. You also have insertions. So you see further down, so the 8th sequence has a G inserted in the position next to deletion. So you typically have more than a few insertions. So this is just a particular case that was mostly deletions. So now what we did a two-step process to introduce the PUF, and in the next 2 slides I'll basically tell you how we built a PUF, and then I'll wrap up my presentation. So the next slide, which is slide 13, essentially contains the protocol. So you start with a single cell line, a monoclonal cell line, that is each cell, they're all the same. The first step is a step of inserting the library of barcodes. So these barcodes are 5 nucleotide long. In our case, it were 5 nucleotide long, but they can be longer. Each position in this barcode could be any one of the 4 letters, so A, G, C, or T. So these all possible combinations of 5 nucleotides, essentially, right? So we used CRISPR to introduce these barcodes. And this is a more sophisticated editing process, which requires some antibiotic selection and it's based on homologous recombination. But in order not to spend too much time on this, so this is a step that is established in the field. So essentially, after this step, we get a barcoded cell line. So essentially, each cell now contains a unique barcode. So there's basically 5 nucleotides that are unique. Now, what we did was in the following step, we introduced errors very close to the barcode using the approach that I mentioned just a slide ago, using non-homologous end joint repair. So we cut in the proximity of the barcode, and that introduces indels. Now, when you're done with this process, you get a puffed cell line, which contains barcodes and indels in the same, basically in the same location in the genome. What you do then is you perform amplicon sequencing, deep sequencing of the particular location, and then you create a 2-dimensional mapping of the barcodes and the indels ranked by frequency. So if you go to slide 15, and that is essentially our path in slide 15, so you'll see that 2-dimensional mapping of the barcodes. Barcodes are on the x-axis and the indels are on the y-axis. You see that they're ranked by frequency. On the top left, you have the most frequent combination. On the bottom right, you have the least frequent combinations. And you see that essentially each one has its own distribution profile, the barcodes and the indels. You see how the indels look like. Now, what is key is that, and this was our hypothesis, and we proved it using 3 different cell lines and tens of different engineered PUFs, was that this particular process, this cyclic process into introducing barcodes and then introducing the indels, results into 2-dimensional mapping that essentially is a PUF. What does it mean? If you go to the next slide, if you follow the same process in 2 different cell lines, you will get a frequency between the indels and the barcodes that is completely different. And you can see these are 2 PUFs with the frequencies, and they look qualitatively different. Obviously, we did not stop at just a qualitative step, so we performed quantitative analysis. So we used metrics such as total variation distance or Brake-Curtis distance to calculate distance between 2 paths. I'll give you one more slide, slide 19, which summarizes, I think, nicely the difference between the PUFs and also points to the robustness ideas. So if you look on the left side on slide 19, there is a PUF11, PUF11FT. So this is the freeze thaw sample. So PUF11 is one PUF, and then the freeze thaw is a sample that after it was established, it was frozen, shipped to a different lab, thawed, and sequenced. Between these two, as you can see again qualitatively, the full PUF, they look very similar. If you zoom in on the top 5 by 5 matrix on the top left, you see the individual positions, the frequency of the barcodes and the indels are extremely similar. We also have a PUF11R, which is a repeated sample, just the sequencing was repeated, so it was done twice to account for just sequencing differences. Right? Now, as you see, the PUF is extremely robust. So once you build it, you can ship it to a customer. The customer will sequence it, will get exactly the same information. Now, in contrast, another PUF that was built is essentially qualitatively and quantitatively, it's different. It's extremely different. So we can safely differentiate between PUFs. Now I'll go to slide 23, which is a summary and a comparison essentially with this plot of our technology with other technologies that are used for cell line characterization. So we've shown in our recently published paper that our approach, the protocol that I mentioned with the barcoding and the indel insertion, is the only one that produces unique, robust, and unclonable signatures. Essentially, I just spoke about the robustness. You saw an example. It was just between two cell lines of the uniqueness. Unclonable because one cannot really build the cell line. A cell line that carries a particular frequencies, that is as close to impossible as it can be. Now, if you compare, let's say, other technologies that are used for cell line characterization, karyotyping or SNP mapping or just barcoding, none of these technologies can satisfy all the three PUF criteria. So I'll give you an example, SNP mapping. So you can say that, let's say you have a cell line that carries a set of mutations, different mutations in the genome. So the cell line, you can ship it to a customer, and the customer can perform whole genome sequencing. And indeed, the result will be that he will get a particular SNP, so that it satisfies the robustness criteria. Additionally, you could potentially say that it is unclonable, right? Because if the cell line has tens of mutations, then it will be close to impossible to actually introduce them one by one, right? So two of the three criteria are satisfied, but uniqueness is not satisfied, right? Because a company cannot sell the same cell line to another customer and have a unique one-to-one relationship, cannot perform provenance attestation. There is only one clone of that particular cell line. Same with the others. So we can go one by one with the barcoding, with indel generation alone. None satisfy the three criteria that are essential for a PUF. So with that, I'll stop. Again, we think that this is an exciting technology. The provenance attestation is a first step for us. We are now looking at ways to lock and unlock information and functionality using PUFs. So essentially, you use the technology to enable a secondary function, right? So it works as essentially as a lock, and you have a key to unlock some functionality that it is unique, right? So, and we're in parallel, we're moving into establishing an entity as a company that will do that puffing for as a service for companies, and then we'll see how things evolve. So, but we feel as a group here that there's opportunities in further science and technology development, but also on the commercial side. So I'll stop here, and I'll be happy to answer any specific questions or general questions. Thank you for your attention.
Yeah, thank you so much for this really interesting talk and for sharing the technology you developed with us. I think it will be also really important in the future. Let's say you 3D print an organ or some tissue, also for the recipient to be sure that it's actually, the product they paid for and not just some imitated cheap, I don't know,
Exactly.
vegetables.
You're exactly on target. We recently had a discussion with a company that is exactly interested in doing what you mentioned, 3D printing organoids, and they worry about that aspect, the authenticity. So this is what the puffs are used for in the semiconductor industry. Exactly, authenticity, verifying authenticity of the chip, and we think it could be used in the same manner in 2D and 3D cell lines. Yeah.
So let's say somebody orders their organoids. How easy is it to break the codes, basically? Like, how is the readout of that code, and is it somehow unique for every, like with everything else, people try to break it, imitate it and whatnot. Like, how easy
Yeah.
is it to
So
replicate?
we're not there yet at the organoid level. There are differences in how you would be able to get information out from an organoid. In principle, we've done we've answered this question for a 2D culture, cell culture, and in our case for a for just a standard cell culture, it's impossible to just rebuild the path. So you cannot clone it from scratch. You cannot build it. In an organoid, we have a couple of ideas, but again, it's just something that is completely fresh. The it could potentially be that you have a heterogeneous mix of cells, and so you have your organoid, and you have cells in the organoid that carry the path, that are potentially engineered in a way that you can isolate them or lyse them, and so then then they leave the organoid intact, but they release their contents, and you grab their contents and sequence them. You could also think about cells that are part of the organoid that secrete that information. Again, this is something that is for us in the works. It's nothing yet published.
Yeah, because my question would be, where do you put those codes exactly, and how do you ensure that it's a safe place to put them where they don't screw up the cells? But we had a guest speaker here from Church's lab. He's a postdoc there, and he's working on safe harbor gene sequences. So he's looking for places in the genome where it's totally safe to put some information and it wouldn't lead into cancer or some other so yeah, I don't know
Yeah.
if that would be helpful.
Yes, so we use safe harbors. I mean, we used safe harbors for this project. We use the standard safe harbor that people use nowadays, the AAV locus. There's a couple of other well-known and characterized safe harbors. The Church Lab is obviously spearheading some of these efforts, and they've been using these safe harbors for a while. Obviously, I mean, it depends on the application, right? So if you want to build a cell line, you want to engineer a CHO cell line that is producing some antibody or some other compound that you're interested, then it's not critical that you're only looking at the yield, right? And if the yield is okay of the cell line, and then you secure the cell lines, you maximize the yield, and you secure it with a PUF, then you've solved the problem, a problem. It's different for medical applications, for therapy applications. We don't. I don't know if I don't want to say no, but I don't know if this directly applies to gene therapy applications. So we don't view these paths as means to improve gene therapy. Potentially down the road, you can think about paths if you think about paths as a way to lock information, which is something that we're currently working. So you can potentially have a fragment in the genome which is secured by a path and nobody can access it. So it's epigenetically silenced, and you can put a number of provisions there to reduce access to it, or sequencing a particular location. And so then maybe this particular location carries a drug or something that you wanna, or an editor, gene editor, let's say. So you've stored in your genome, and I'm just brainstorming here, right? So you've stored in your genome Cas9 and you have it dormant, and you have it locked, and you have a unique key to unlock it that is personalized for you. Only you can do it. Then I can see, people may start thinking about worrying about potential, issues and just off-target effects and other effects that you do not account for. There's obviously lots of things that need to be done to do quality control and so on.
Yeah, thank you for answering my questions. And yeah, we have more people on the stage. Dr. Shah, Dennis, Nas, Joyce, Frank, Kyle. Welcome, everyone. Go ahead with questions.
Yeah, thank you so much, I mean, for sharing fascinating work with us. My question from you is about, because you just explained about the mechanism as well as the gRNAs and everything, and how much is the potential of the system for expanding, for example, with considering the multimerization, or as you know, that is a complex formation, as well as the multiplexing. So I was just wondering, do you have any result out of your experiment into it?
Your voice, the sound fluctuated a bit, and I missed the kind of the middle of your question.
Yeah, I mean,
So
the main emphasis was about the multimerization and formation of the complex formation that you know, and multiplexing, which we know that is a different target in the same cell. I was just
yeah,
wondering if you have any, I mean, result or numbers about the numbers of the effectors, possible effectors on a system for expanding the system.
yeah. So in our particular case, I mean, we did 3 cell lines for the particular paper. We use for these cell lines, we use for all of them the AAV1 locus. We have a particular location in the locus, a particular guide, a particular target in the locus that we've verified in the past that does not cut in any significant significant other location that has any significant off-target effects. So we were quite confident that there were no off-target effects. Now, in terms of multiplexing, in our case, we are essentially we're doing editing on a population of cells, right? And each cell does its own edit. So this is essentially, it has its own resulting into a different indel essentially. And so you're in one step, you produce a population of cells that contain sequences of barcodes and intels that are different to each other. And obviously, when you move up to hundreds of thousands of cells, you have then cells that carry the same barcode and intel. So then that's why we're working in the end with frequencies. So that is the baseline process. We're working on an idea that has to do with multiple cuts in parallel as well. So you don't have just a single cut, you have cutting in different locations. And so then you combine the information from two NHJ events. Yeah, so
And how many gRNA you use for fusing?
Oh, in our case, it's just it is in this paper we have a single guide RNA. In the work that we're doing now, we use a couple, so it's just two locations. In principle, you can use more. But it's not. We don't see the. I mean, we get enough diversity with one. We're using two for a different scenario that we have in our mind. So one gives you, for us, gives enough diversity to satisfy the the uniqueness essentially, right? So what you want is two cell lines that were cut to have a resulting frequency that is sufficiently different. Yeah, but you can
Thank you.
multiplex. You can, I mean, you can essentially the technology. If I can take it a step further, I mean, people are doing screens with CRISPR, right? So it's not. You can use a library of guides. So we used a library of guides to multiplex edits on microRNA targets a few years ago. So, and there are a number of screening, CRISPR-based screen papers. So I'm sure you've seen. Thank you for your question.
Leo, thank you so much for this really interesting presentation. I was curious about something sort of probably very simple, but I was curious how it is that the guide RNAs lock into specific sections of pathways. Is it each one is keyed to a different part of a genome, or how does that work?
So how do specific guide RNAs
Yeah, like there
lock
are
on?
several types of guide RNA. Is that correct?
Well, I mean, there are many different Cas proteins, right? So its bacterium has evolved to have kind of a different, slightly different Cas and a slightly different guide RNA structure. So there is the most well-known Cases are SP-Cas9 and SA-Cas9, and they're different. The proteins are different, and the corresponding guides are also slightly different. The structure of the hairpin is different. There is now Cas12 and Cas13 that these other proteins are targeting DNA and RNA respectively. So this is it, because of the huge diversity in bacterial species in bacteria, then they've evolved and they've evolved for a particular environment, and they've evolved their defense mechanism, which is their phage defense, the defense mechanism against phages, right? So it's part of their adaptive immune system. And so they've evolved these proteins and guides for their environment. So in any case, what we use is a single Cas9, SP-Cas9, and we use a particular guide that is recognized by that particular SP-Cas9. So we don't use different guides for our experiment, right? So the only thing that you change that you would need to change in our experiment is the target location. So, but essentially we don't change that either, right? Because we have a target on AAV1 on a particular safe harbor, and we cut there. That is where we introduce that. We let the cell then fix itself and just introduces errors, and that's it. We have a different guide to cut the genome in a slightly different location, a neighboring location, to introduce the barcodes with homologous recombination.
Understood. Thank you so much.
And I'll be very happy if you have additional questions or secondary questions, or if I did not really, answer your question exactly, I'd be happy to respond to email. So and open the discussion. So my email is my last name at utdallas.edu. So feel free to just shoot me an email.
Oh, hi, Doctor. Yeah, thanks for sharing your interesting research with us. So I have a quick question regarding the application of this important technology is as you the analogy you made with the computer um, encryption as well. Right now the application is towards cell line specifically, or are you also thinking of applying to larger areas, other broader scenarios?
We are starting with cell lines. So we have results for cell lines for 2D culture. So um, essentially, any cell line that has been engineered. So you can, again, as I mentioned, there are tens of companies right now that have engineered cell lines that have introduced mutations of interest to particular genes. So if you go to ATCC, you'll see that for a particular cancer, they have a number of mutations that carry a particular mutation. KRAS, for example, KRAS mutant, um, um, I don't know, 13, what is it, there's certain KRAS mutants or 53 mutants. So these cell lines, some of these cell lines are quite expensive. So they cost about $10,000 to $20,000. So we think that it would be beneficial for the companies to be able to secure them and be able to do transactions one to one. So basically sell a single cell line to a single customer and ensure that only this customer can and will use that particular cell line. So it will maximize the profit. But at the same time, as I mentioned before, I think this is something that it took a little while to be picked up in the semiconductor industry, but now it has become commonplace and has solved a number of problems related to security and provenance attestation. I think with the developments in cell line engineering and 2D and 3D, there will be definitely a need for this type of technology. Another issue that I have not mentioned, and I think it's quite important, is that there is significant kind of mislabeling of cell lines. So it's very often that you see basically labs that use cell lines that think that they're one cell line, but it's a completely different cell line. Or that it's some cell line that's evolved to be different. So this our technology, this technology, or these types of technologies similar to what we built here, can help kind of um, improve the quality of research. So if there is, if you thaw a cell line, you sequence a particular location, and you have that unique information, you know essentially where it comes from, who built it, if he performed quality control, and so on.
Thank you. So, if I may, another quick question regarding the. I assume that there's some mathematics behind this research as well. For example, the RCA, like in computer science, the
Yeah,
crypto encryption.
right.
I guess here you're using different bits of information units bits here
Yeah.
specifically. Any could you share a little bit? Yeah, thank you.
Yeah, so we use simple metrics actually here. So we use a so-called total variation distance. So essentially, we convert these two-dimensional matrix. So we convert the two-dimensional matrix to a single vector. That is basically we combine the barcode and the intel, rank them with respect to the frequency, normalize that, and then use that single vector and then start comparing vectors to each other. And so we use Bray-Curtis distance, which is just a simple statistical tool, a formula essentially to compare vectors, the distance between two vectors, and we use total variation distance. And these are two approaches that we've used. The total variation distance against compares everything, creates a mapping of all data that you have. The Bray-Curtis compares vectors one to one, and so it gives you slightly different perspective. But essentially, there is also that both work. The technology in both cases, in both downstream quantitative tools that we use, they worked great. We are also now have started looking at machine learning as kind of looking at the this two-dimensional mapping with machine learning tools to essentially create, extract features and then compare features. So I think there's lots of opportunity there in that front. It's a good question. I think it's kind of underexplored at this stage, given essentially that this is a new technology as well.
Thank you. I think this is a very interesting application of many exciting new technology from science and research. Thank you, Doctor.
Thank you.
I have a quick question. And thank you, by the way, for a very interesting and viable conversation. Thank you, Katrina. Do you have in mind any uh, applied application for this technology, for this innovation that you lectured, whether for diagnostic purposes or therapeutic, that can actually be commercialized, or is it still pure research?
Yeah, so we are. Yeah, thank you for your question. We've moved in establishing an entity, a company here in an incubator that is our first goals is to provide a service that basically that barcoding, that puffing of cell lines. So if a company builds a particular cell line that wants to protect from uncontrolled distribution, we can ensure that the particular cell line um, will be again puffed and stored in a way that they can control who uses it, and they can control their IP. So that is the first immediate opportunity, I think, that is there. There is a need for solution to it. So we see that there is an opportunity there. We've had a couple of companies contact us for licensing the technology. We decided to pursue this ourselves, so we'll see how things evolve. Beyond that, as I mentioned earlier, we think that there is, again, opportunity to use these PUFs as a lock and unlocking mechanism within anything that this might, entail. So we're working again. There's interest by this company on organoids, and so they have clearly they've identified some commercial opportunity, and they want to secure the organoids. There was interest in from the perspective of data storage, so DNA-based data storage. So you have information that is stored in the DNA, and you can secure it, you can lock it, and you can ensure that only the person that has access, the key to the PUF, will access the information. But we're starting with just the first step for us is puffing cell lines. And we think that, considering the size of the market, there is, and the developments, there's now companies that build cell lines using genome editing. There's tons of companies that build custom cell lines. And so I think the companies will want to protect their product in their times. So we'll see.
No, it's wonderful. Thank you for the elaboration. You mentioned IP, and obviously your business concept is very viable, and indeed you should protect it. Have you filed for patents? Have you protected your intellectual property? Because once you enter into the commercialization, this is a jungle outside the laboratory.
Yeah, we've filed through the university. It's been now more than a year that it's been filed, and so I hope that this is finalized and that we have that we can exclusively use the technology. So
Awesome. Of Hairstone.
thank you.
Yeah, Joyce, did you have a question? I think you didn't get a chance to speak yet. She might be away from the phone. I wanted to check in with you how much time you still have left for us, or if, because we are 12 minutes over an hour, so yeah, you might want it's probably getting late for you, so I just
I
wanted.
think this was extremely fun, and you've asked excellent questions. I think my ability to answer smart will gradually diminish, and so
Yeah, I understand. So it's
yeah.
been a long
So
day.
I'll be very happy to continue the discussion by email. So you guys, if you have any question or feedback, suggestions, or ideas that we can pursue jointly, then I'll be more than happy to respond. Again, you can get my email from Katarina or directly my last name at UT Dallas.
Yeah, thank you so much for presenting and for answering all of our questions again. And I think, yeah, we wish you all the best for your company. I think this is really important, especially in the future. I worked for consulted for like 3D printing of organs and things like that. I think also there, like just for quality control and for the safety of the patients, that it's actually the right cell lines and not, fake cell or just the wrong cells is really important, because if you get an organ that's not the right one for your immune system and so on, it's really bad. You know, you
Okay.
can. So yeah, thank you for doing that, and thanks for sharing. And we wish you all the best for your startup. Thank you.
Thank you. Thank you for the opportunity again. Thank you for your time.
Yeah, and thank you everyone for coming and asking questions, supporting. And yeah, if you like discussions like these, follow the club. We will have this week 2 more talks. We will have Dr. Moseson coming tomorrow. She's a public health researcher, and we'll talk about the public health, how it's affected by abortion, by the new abortion laws in some states, and how abortion attempts without clinical supervision, how bad it can turn out. So it's kind of the bigger picture, and then also stories. And then on Friday we'll have Dr. Araujo, and talking about how an inner ear can tell us of the origin of warm-blooded mammals. Will be a really interesting talk. It's really interesting how the inner ear is so important for evolutionary biology. The previous guest speaker here talking about how he used ancient DNA from the inner ear to trace back who were the first seafarers, and now it's to see how warm-bloodedness evolved. So the inner ear is kind of very important. So keep it safe. No, I'm kidding. Okay. Okay. Thanks everyone. Have a good night, good morning, good day wherever you are. And yeah, I'll hear you all back soon. Thank you.
Thank you. Thank you, everyone. Thank you, Leo.
Thank you all. Good night.
Okay, I'll close the room in 3, 2, 1. Bye everyone.